welcome for this Global Bioimaging Spotlight seminar. We're very happy to have you all here and today to listen to Kildare. We'll come back to Kildare in a minute before we start, and also to give a little bit time for those who are still joining. I would like to run by you also a little bit some updates and announcements from the Global Bioimaging community. Johanna, you can go to the next slide. I think we start for, I'm not sure how many new people we have here, but looking at the Global Bioimaging International Network of Imaging Scientists, core facilities and infrastructures. So there's quite a lot happening, and we welcome you all to look into what's happening within your own, as well, your own region and network partner, but also within the Global Bioimaging. On the next slide, I think the first thing we want to do is to Johanna, you can go to remind you so that this seminar and several of our activities and outputs come very much from the working groups within Global Bioimaging that not only work on international recommendations, but also organize meetings like this one. So you're very welcome to explore in our website about the different working groups and see if you'd like to join one if you're not yet part. Also, if you have any questions, don't hesitate to reach out to us to know a little bit more what the group is currently working on. And I think the next slide talks exactly about, the newsletter. For those who want to sign up for the newsletter, we can put the link on the chat as well. About that, what the Career Path Working Group is doing after the publication on the in recommendations for imaging scientists, careers and core facility. One of the outcomes is what we're starting working on is this webinars and this GBI spotlight, where we every time spotlight a different success story or a different regional story and career paths topics. But as well, there are several other actions. So the group has grown from, I think, 40 participants to 70 registered members. And the next one, you can go to the next slide, Johanna. One of the things we're working out right now is we're running a survey, and we are working also on some guidelines for authorship and acknowledgement of imaging core facility staff and imaging science contributions and publications. So we would like to invite you all to fill out our survey, because this will definitely inform us and help the group and the global community to develop, to promote guidelines and to advocate for a stronger recognition. Although some of imaging facility staff has started to be recognized in acknowledgements, what we're looking at is the recognition in authorships, and not only in the authorship for publication, but in where in the authorship as well. So we welcome you all. I think we can put the link also in the chat, but if you didn't have time for the QR code, please do take a picture. And finally, the next would be to remind you some upcoming activities for this year. So we welcome you all to join our annual meeting this year in Montreal, in Canada, with a focus exactly on how to build resilient and sustainable infrastructure, imaging infrastructure ecosystems. And we'll meet in Montreal in October, and the registrations will open very soon. So next week, the registrations for this meeting will open. What is already running, and you can click again, please, Johanna, is the registrations for the core facility management course that will happen right before. So if you see here, we'll first start in Montreal with the core facility management course organized by CLAB, then the annual meeting, and then follow up by the Bioimaging North America Community Congress. So you're very welcome to apply right now for the core facility management course and travel grants. Travel grants application closed by end of April. So we have a couple of days for applying to that. We definitely support the most participation possible in these events. If you have any questions, please also let us know, reach out to us at any time. And you can click again. I think in terms of announcements, we also will have very soon, so by mid-May, we will start, or GLAB will open the next international job shadowing call for imaging scientists that work in core facilities and that would like to spend a certain period of time in another core facility from any of our partners. So at this stage, Global Bioimaging composed of 13 official partners and encompassing more than 64 countries. So for those who would like to apply for the job shadowing program, please stay tuned and keep in mind to check our website in two weeks, more or less. And as the same way, the next slide shows you the reminder for the Imaging for All initiative. For Imaging for All supports not only with grants, not only training in imaging, but also user access for different facilities or other departments for accessing technologies you'll not have available in your institution. And you can apply from training with 5,000 euros per application, or access or Pro 10 and 20. And the first call was a success, just closed this year, and the next call is going to be coordinated by Denise, our colleague, and will open on May 12th. So please stay tuned for these opportunities. And the next slide is to bring you back all here for why we are here today. And I'm very, very honored to present Kildare Miranda, who's coming to us from Rio de Janeiro, from the Biophysics Institute and National Center for Structural Biology and Bioimaging. Kildare is not only a very active member of the Career Path Working Group as well, but also at LABI, at the Latin American Bioimaging. So Kildare, we're really looking forward for your talk and for the discussion afterwards in understanding some pathways and detours of an imaging science in Latin America. Thank you so much for being here, Kildare.
Thank you so much, Yara, for the introduction, and also to Johanna, you, and Sophie for the invitation to talk about what we're doing here. So we'll try to keep it very informal, right? And just to tell you a bit what I've been doing here and the next steps that I plan to follow here. So let me just try to share my screen here. Is that okay?
This is now the presenter view. So if you have to swap the displays.
Okay. Just 1 second. Is it now?
No, but if you go at the top there on the swap displays.
Okay.
Yeah, now it's perfect. Thank you.
Okay. So, so then as Yara already announced, Yara already mentioned, we talk about the pathways and detours that I had here during my career. So just start briefly with my with the things when I started, right? Since I started my studies, so I did biological sciences. I finished in '98, so I'm a bit old. And so I did my undergrad studies at the North Fluminense State University, and back then and up to now in Brazil, we have a very strong program called Scientific Initiation. So we are within the labs since the beginning of our undergrad studies, sorry. And I was already committed to projects that involved microscopy at the time. So this was a new university focused on science and scientific activities. So we had a very strong, at the time, very strong microscopy part, and I was already involved with that. Then I finished my studies and I did my master's and my PhD at the Biophysics Institute here, where I am now. And with a short sandwich period at the University of Illinois. And here at the Institute, we have a long tradition in doing microscopy, especially electron microscopy. So the first microscope here was installed in 1952. I didn't use this one. But it was a relic that we had here at the Institute. This microscope used glass negatives. I don't know if you have seen one. We have a bunch of the glass negatives here, very interesting. Then after that, a series of instruments were installed at the Institute, and we had this long tradition in electron microscopy, and I've been involved with that since my undergrad studies. So then I did a short postdoc at the University of Constance in Germany. Then I came back to Brazil, moved to the USA, to the Center for Tropical and Global and Emerging Global Diseases. So I worked with infectious diseases since the beginning of my career. So I did a postdoc there with Roberto do Campo. And in 2006, I became an adjunct professor at the Biophysics Institute here at the Federal University of Rio de Janeiro. So my work, I'm not going to talk about the science that I do here very much, but just briefly, just to let you know, my work is focused on infectious diseases. So I work mainly with trypanosomes. I've been working with toxoplasma, but most of my work with apicomplex and parasites now it's with malaria. And we are mainly interested in transporters of ions and osmolytes and biosynthesis of crystals, and a few structures that we are interested that compose the acetylcostome, contractile vacuole complex, food vacuole, hemozoin crystals. So all these structures we study with the different light and electron microscopy techniques. So just very briefly, this is one of the structures that we study. The acetylcostosome is basically a bag full of ions with a lot of pumps, transporters, ion channels, and we are interested in these ion fluxes that happen in the organelle and how this translates into infections, into pathogenesis, right? And we are also interested in the formation of polyphosphate polymers within these organelles and the functional role of these polyphosphate polymers. Very briefly, if we look at the microscopes and at the acetocostomes in electron microscopes, we see these black dots, right? Especially if we use a microscope with an energy filter. So we see these organelles, these black dots in different species that we can quantify, we can measure, we can do a lot of things with them. We can analyze, if we use an analytical microscope, we can analyze the elements that are inside the organelles. We can change the composition of the organelle with different stimuli, the composition, the structure. So it's a very active organelle. It's very interesting for us from this point of view. So the main motivation to do that is that it's because the parasite alternates between different hosts, so mammalian hosts and the insect vector, and the parasite crosses different tissues and different environmental conditions such as temperature, nutrition, osmolarity, and other things. And I'm mainly interested in osmotic stress that these parasites face during their life cycle. And for that, we study also the contractile vacuole complex and the biochemical pathway that leads to different osmotic responses by these parasites. So we've been characterizing the structure of the contractile vacuole with different EM techniques. This was a work done by Ingrid Augusto. So I'm just mentioning now the recent work. This was published last year. Also, Wendell was also working with the formation of molecular condensates within the organelles that we study. So the main focus of the group is in infectious diseases and how the structure and the function of these organelles can translate into pathogenesis. For instance, the organelle is full of polyphosphates, and we are studying the role of polyphosphate in vascular permeability. So if you think about one parasite that needs to cross different vessels to reach different tissues, it's important to have a storage of molecules that can induce vascular permeability. So this translates the structure and chemical work that we do into pathogenesis. So, but that's all that I wanted to talk about science. I'm just focused now what I've been doing related to images and how I've been working with the community here. So then right after I got my position here at the Institute, after a brief period at the Metrology Institute, where we also built an infrastructure for microscopy characterization, both for material sciences and also life science, I came back to the Institute to work at the Senabio project. So we started writing the first grants in 2008, but we got the first microscope in 2010, but the building was ready only in 2015. So it was a long story to start building Senabio. So the aim was to establish the National Center for Structural Biology and Bioimaging, capable of studying a biological phenomenon in mesoscale, from the atom to the whole organism, right? This has been our dream since ever. And as the first chance that we got, we started to build this infrastructure together with the colleagues from the Biochemistry Institute, as well as colleagues from the from the Biophysics Institute that are more focused in animals. So the whole idea was to do structural biology of macromolecules with NMR, X-ray, cryo-EM, molecular modeling. Then on the other end of the spectrum, bioimaging of small animals. And we are here in between. So the microscope have this responsibility to bridge between cells, organelles, molecules, atoms, and the other end of the scale, cells, tissues, organs, and the whole animal. So for that, this is what we already have in Senabio. So we have a collection of magnets from 400 to 900 megahertz, and also some infrastructure to do prototyping of molecules. Also, in the other end of the scale, we have 7 Tesla, MRI, also PET scan, bioluminescence with CT, echocardiography, all these things, different methods to study animals. And then we come to the microscopy site. This was where I was involved since the beginning. So we built our light microscopy facility, which is in the 2nd floor of the building of Senabio, where we have super resolution, turf, FCS, FLIM, optical tweezers, so a number of instruments to do light microscopy. We're upgrading now to a new multiphoton microscope, a new high content analysis from tissue agnostic. So we're always trying to evolve here. Also, we also have at Senabio a unit for drug discovery, so for high content analysis from multiparametric studies for different cell markers. So this has given us a lot of work to put together. And then we come to the microscopy, electron microscopy facility. So then a brief comment. I was mentioning to Johanna that at Senabio, it was a very, very hard time to put these things together, because we have to deal since the construction of the building to the installation of the instruments, training technicians, writing grants. So it was a lot of work with a very, very little administrative support. So I had to do a lot of things in our own way. Because this was not well embedded in the structure of the university. So these facilities that concentrate instruments, it was something that was not embedded in our framework. So it gives us a lot of work to put this together. So then we have now a number of TMs and SEMs with variable capabilities from regular sectioning to three-dimensional capacity. And then in 2023, we started building a new high-end cryo-EM facility, so both for single particle analysis and tomography. So back then, we only had this entry-level old cryo-EM. I don't know if we can say this is our entry-level anymore, because now entry-level are the new instruments without the whole capabilities of the high-end ones. But we have old instruments where we could fit side entry cryo holders and do basic cryo-EM, even tomography, but not with high resolution. Then we started with the project in 2022. 2023, you got the grant to the end of 2022, we got the grants, we purchased the instruments, we installed the building was ready in the end of 2023. And we installed our first trios with the direct detector, with the whole package for cryo-EM, both for single particle analysis and cellular tomography. So to do that, we have to construct a new building. Again, the whole, this is like this things with these games with the children, we roll the dice and we get back to square one. We had to deal with these creatures during the construction of the building. This was this was not the only time. A few times they came from the neighbor regions. So, but then the building was ready, and we got to do the first experiments with our trypanosomes. It's an open facility, but as we were the first ones to establish the technique and start working with the instruments, we wanted to study cells that we were familiar with. So started with trypanosomes to do doing single images at the old instrument, and then priority with the new microscope. So we studied the structure of the acidocososome, which was our main model, so we could directly see these phase transitions within the matrix of the organelle, which was really, really nice to see that there are really something that we were hypothesizing for a while. Now we could have the proof that we have phase transitions within organelles. This is the first time that we're seeing that. We prepare some lamellas from our parasites. So I mentioned that I'm interested in the contractile vacuole complex, and we have a number of proteins that we can identify in the contractile vacuole complex, and we have cells that are transfected that express the GFP linked to RAB11 or AP 180. And with that, we could do cryoclam, we could do freeze down the samples, do fluorescence microscopy, and then prepare the lamellas on the samples, on the cells that were providing this signal. And then we did tomography, and we identified the number of candidates that we're now proceeding with subtomogram averaging to see which proteins are involved in the contractile vacuole complex. So this was a very, very I think that we reached a certain maturity within our center, because the scope was ready in June, and we already had the first results last year. And in the beginning of this year, we already wrote the papers. We will have a couple of papers out. Also could see for the first time the polyphosphate molecules within the acidocososomes within the organelles. We can actually see these polyphosphate molecules, which was very interesting for us to see that. So as I mentioned, a couple of papers already out this year using the instrumentation that we installed. So I'm really happy with that. So the group was really fast moving forward with our work here. So, but then back to the community work. So in 2016, I was always involved with the Brazilian Society for Microscopy and Microanalysis. In 2016, I became the president of the society. And also in 2019, I became the president of the CIASM, which is the Inter-American Committee for the Society of Microscopy. So it gets takes from Canada to Peru. So all the foreign societies are affiliated to CIASM. And it was through these communities that I got to know. We organized a lot of congresses in different places. I just put it a few here, but I got to know better the communities, their needs, and what we could do for them as an institution, as an organization, as Brazilian Society for Microscopy and Microanalysis, or CIASM, and also as an institution, because we have the instruments, we have people here, right, that could help different groups. So through the work with the SBMM, we did the landscape analysis of the Brazilian microscopy community. I already presented this paper a few times, so I won't talk about it. But this, I think it's important to mention that we should quantify things that we already know. So everybody knows that in Brazil, we have this asymmetry, that most of the investment comes to the south and southeast of the country, but this reflects in a lot of different aspects that you want to quantify, and one of them is the investment in microscopy. So we have a fairly good installed base of instruments in the country. We have around 700 microscopes, but most of them concentrated in the south and southeast region. So, and because of this, we then moved to Manaus. We wrote a grant together with Vanderlei. We submitted to FINEP, and we got the grant to install the CMA Bio, where we have Jander, Aldenora, Jessica, that some of you know, that are working there and keeping things moving there. So we're really happy with the installation of this new center, which serves the whole region. And there's a lot of people working there with them. And also we have the very close relationship, a very close contact. So from time to time, we go to Manaus to give some lectures, workshops, courses. They come to do training. So it's a very, very nice initiative that we have. And one of the consequences of this of such type of studies that once I got, you know how our careers are. So here I can't have the luxury to be doing only one thing, right? Only taking care of the center or only doing my scientific work. I have to do everything, otherwise I'm dead. Scientifically, with the federal agencies, or if I don't do well, manage well things that I'm responsible here, then I'm not in a good position in my own institution. But once we have this under control, and I think I have this under control now, we started to look at other possibilities to help our community. And one of the things that we got to know with these studies is that we have this big symmetry in the country, and also that hospitals are not used to use electron microscopy to do to reach their diagnosis with pathology. Right. And then we wrote a project. We submitted to the Ministry of Health in Brazil. We have this centralized, unique system, which is public, it's free. Nobody has to pay anything to use it. And it's very well organized, has a lot of capillarity. And then I wrote a project, contacted. This was something that I learned in one of our workshops together with the GBI and CZI, that if you have an idea, you should pick up the phone or write an email, talk to the director there. So I did that, and I convinced them to fund a project to do telemicroscopy applied to structural pathology. So the same principle applies. We have this asymmetry in the country, but we have also restricted access to pathologists to the equipment or to the network here. And this is mainly because they are not used to do EM, or it's too difficult, or it's not in the frame of their hospitals. So we started this project with them. So for a number of pathologists, just brought one here, for instance, respiratory diseases, with primary ciliary dyskinesia, which is a rare disease, but you only close the diagnosis if you do EM. So the idea is to work with different hospitals, which is what we are doing now. Work with the hospitals, so get molecular data from proteomics, from genomics, proteomics, genomic analysis, biochemical analysis, and also now with pathology, light microscopy, and electron microscopy. The idea is to have interoperability, so to have metadata associated to those images, so that we can track what happens with each patient. So the idea is it's not now it's not a program to do a massive assistance. It's just to test a model and establish a sustainable operational model for diagnosis using electron microscopy. So the idea is to ship the samples to the centers. The physician on the other side of the screen can join us. We can have a joint session in the microscope, take pictures, and close the diagnosis. Or we can train the technicians. They can take the pictures, upload it into the cloud. Then the physician, the pathologist access the images and give the diagnosis. So this is more or less what we are planning. So the idea is to develop a strategic telediagnosis plan to apply to the model on a national scale once we are finished with that, and test also the application of machine learning and AI to assist telediagnosis by electron microscopy. So we also, as we do a lot of 3D EM, we're also testing 3D technologies to do digital dissections of cells and tissues to identify planes that are favorable to diagnosis. For some of these diseases, you have to have a perfect plan to see the structure that indicates the pathology. But then once we're able to get three-dimensional data sets, we can do it digitally. So this is something that we're testing. So, and then again, I think that my thing now is to try to build networks to connect people. So once we have the high-end instruments here in Rio, and we also have high-end instruments in São Paulo to do cryo-EM, we started with the idea to build a cryo-EM network in Brazil and then expand to Latin America. And but the main problem that we have is people, right? We have to have the right people in the right places. And with that, we have already a number of former students that were here in the lab that know how to do either cryo or three-dimensional work. They are now in different places in the country. And then now we're putting together this network so that we can, the main idea is to have the national hubs like Senabio, like Senepen and Campinas, which have the high-end instruments, and give support to the regional hubs, either to learn how to do screening, to work with low-end, with entry-level instruments, low-end instruments, and then once they are able to prepare the samples, they could put it in a dry shipper and send it to us, and we can do the high-end work here. So then we are working with this hubs model, and we're helping them to establish the different labs to do the basic work. I'm happy to say that just this week now we got the funds released to upgrade the microscope in Manaus, in the middle of the Amazon. So we'll be able to freeze down the sample, have a plunger, a new camera, side entry cryo holder, pumping station, so that we can take samples from the Amazon. For instance, my personal interest in Brazil, the main problem with malaria is with malaria vivax. And this is a parasite that we cannot cultivate, right, for a longer period. So it's very hard to have these samples in advanced labs. So the idea is to get the samples there. We already have a cooperation with the Tropical Medicine Foundation there, where they are able to get the blood of infectious patients, infected patients, and go through the mosquito, differentiate the parasites, collect the parasites. We can freeze them down and do the screening there, and then complete the work here. And we could do this for other things related to the biodiversity in the Amazon. So I'm really keen to do that, really anxious to see when the instruments will come and we can do that. Also, we are part of a network, an international network, which some of you know and are part of also. So this is a project coordinated by José María Carazo in Spain, which is a cooperation between countries in Europe and countries in Brazil. So the idea was to come from a situation like this where we had informal corporations now with a funded, very articulated networks, and really involved with such things. And also, it was through the CIASM, the work that I was doing in CIASM, that I came across Andrés Camaid and Lia Pietrasanta, and there was already an idea to establish a Latin America Bioimaging Network. We worked together. We've been working together for the past years. Andrés was the first chair of Latin America Bioimaging. Lia was the second chair, and I was the starting now as a chair. We started in Lia maybe no 2 weeks ago, maybe. Started as the chair of Latin America Bioimaging, and we are working we talk almost every day, right, Lia? So it's a work that we've been doing a lot.
Todos los días, every day, yeah.
And with through the Latin America Bioimaging and also our institutions here, and thanks to the generosity of Global Bioimaging and Gleb and the whole team, we organized the facility management workshop here last year. We had a number of speakers from different places. It was really was great. It was great to organize this here. Also, we organized the first Latin America Cryo-EM workshop. So the scopes were running for a couple of months, and we organized a meeting here with Dorit Hanain, with her group from California, and also a group from the Max Planck Institute in Dortmund. Was also really nice. And then we followed with these courses. In between these courses, we organized the lab meeting in Rio, together with the super resolution fluorescence microscopy meeting. We organized here at the university. We had around 300 people in total. It was really nice work. Wow. This is the same slide for those that were in Japan. This is not new, but it's the same slide that I prepared to Japan, because actually this was a very close, right? Japan was the end of last year. So the lessons learned, it never ends. We always have new technologies. We need more funding. We need more space. We need more talented people. We need more. We have more problems. We need more solutions. So it never ends. So this is one thing that if you are going to the imaging business, you have to be prepared. And the collaborative work is essential, but it can be a very lonely process. As I mentioned, for me, it was a lonely process, but with time you have more people involved, and once you get that, you have unconditional support from the community, and then we have more grants approved, more visibility to the funders, more fellowships for technicians, so things grow in the way that you cannot expect. And now the big problem that we have is that we have big data sets to manage and to analyze, right? We need computational power, need software, need people. And the answer to that is community, right? Now, I just moved from Senabio to the Biophysics Institute. So the place where I started my master's, and which was the beginning of everything. So the first microscopes were here. I learned how to use the instruments here. Now I just left Senabio in the beginning of the year, the end of January, and now I'm the director of the Biophysics Institute. So now I have other things to do. I'm still in the imaging business, but because this is my core, right? This is what I do. But now I'm in another it's another challenge, a little bit different, a little bit larger, and a lot of responsibilities. But I think that it's a great opportunity to reflect on what I've done before and apply what I learned to our community here now, not only for imaging, but during or infectious diseases, but dealing with neuroscience, dealing with immunology, dealing with the precision medicine. So I have a lot of other barriers. But guess what? Imaging is embedded in all of it. And I'm happy to say that I can help a lot the Institute with the background that I got with this previous experience. So just to finish, I'd like to thank my group members. So these are the guys that do all the hard work, right? I won't mention each one of them. And also to our collaborators in Brazil and the US, also in Argentina. Also at Senabio, to my group at Senabio, which now is Marcia's group. So the first one here is my replacement led there, was the person that is leading the team now. And thanks also to our funders, and I'll be happy to take any questions. Thank you.